All right, welcome back, everyone. So today we're going to get into chapter 10, which basically we're going to look at meiosis. Okay, so we talked pretty extensively about mitosis, where we have our somatic cells dividing and basically making identical daughter cells from them. Meiosis is a little different because we're working with our gametes. We're producing new gametes that are not identical to each other. This is where we get a lot of variation in our genetics. So we're going to talk about that and make sure we understand uh, what chromosomes are and why we have 23 pairs of them. Okay, so we're going to get into that today. So let's start out by discussing about our chromosomes and where they came from. So we inherited them, of course. And so this term heredity is defined as the transmission of traits from one generation to another. So you acquired your traits from your parents, and your parents acquired their traits from their parents, and so forth. So basically, through the passing on of our genes from generation to generation, we are transmitting our heredity, or so transmission of traits. However, each one of us is genetically unique. So even if you have siblings from the same parents, you have a unique shuffling of your parents' genes, and that's what makes you different from your siblings. You don't look identical to them. And so each one of us has this unique combination of our parents' genes. So this leads to variation within our species. And so genetics, we're going to be focusing on genetics for the remainder of the term. And genetics is the study of heredity and the variation that is caused by it. Okay. So in order for us to get into chromosomes, we need to take a closer look at them. So what are chromosomes? We talked about chromosomes in the last lecture being composed of chromatin. And chromatin is basically just a string of DNA with histones, right? That protein. Now, if we take your DNA strand that's found on your chromosome and unwind it, okay, so you just imagine this long string of DNA. That DNA is made up of those nucleotides, the A's and the C's and the G's and the T's. And a combination of those nucleotides, a segment of that DNA, is what we call a gene. So a gene is just a segment of DNA with the combination of those nucleotides, those A's and the C's and the G's and the T's. And so genes are going to code for a trait. So that is our unit of heredity, because you might have genes that tell you to have straight hair instead of curly hair. Okay, so those are segments of your DNA that code for a particular trait. And so we inherit those genes from our parents, right? Now, if we're taking a look at all organisms, remember, not every organism has two separate parents, okay? So we do have asexual reproduction in some of our organisms. So think of, here, this is a type of animal called a hydra. It's related to jellies or sea anemones. And it is undergoing some asexual reproduction. So basically this guy needs a friend, so he grows one. So that bud that will bud off of him will be identical to him. So this is a form of asexual reproduction. So the clones will be identical to the parents. Plants, they do that as well. Lots of different organisms do that. Some plants and animals and other creatures undergo sexual reproduction. So sexual reproduction means that we're going to have two separate parents providing genetic information to form an offspring. And because your genetic source is from two different sources, you're going to end up with unique offspring through sexual reproduction. So asexual, think cloning, sexual reproduction, unique offspring. And in order to get unique offspring, the parents need to pass on their genes to their offspring in a certain unique fashion, which is basically through meiosis, where we're going to be producing gametes instead of a somatic cell like we did in mitosis. So those gametes is just a scientific term for sperm and eggs, okay? So let's take a look at the life cycle. So life cycle is just generation to generation sequence of stages in reproductive history of an organism. So we follow this particular life cycle. We'll focus in on humans and more complex animals, where we are basically what we consider a diploid organism. So what does a diploid organism mean? It means that you have two sets of chromosomes. So you have 46 chromosomes. And there's two sets of them. One came from your mom, one came from your dad. So we actually have 23 pairs of chromosomes because 23 came from our mom, 23 came from our dad. So each human adult is, or kid, doesn't matter, is a diploid organism. So we are diploid, and each one of our cells has two sets of chromosomes, okay? 23 from mom, 23 from dad. However, we can produce haploid gametes. So haploid means half as much. So instead of making a cell that has both sets of chromosomes or 46 chromosomes, we have the ability to make cells that have only 23 chromosomes or half as much. So that is through a process known as meiosis. And so we're taking our 46 chromosomes, we're dividing, making cells that only have 23 chromosomes or one copy of our chromosomes. And it's a unique copy. It's a unique combination of those genetics. So that's where we produce our gametes. So our gametes are produced through meiosis, where we end up having the amount of chromosomes. So instead of having 23 pairs, each one of our gametes has just 23, or one pair of each chromosome. And so that way, when we have a haploid egg with its 23 chromosomes unite with a haploid sperm with its 23 chromosomes, you end up with a diploid zygote. So a zygote is just a fancy term for a fertilized egg, by the way. And so now we have this little baby human. It's just a one-celled zygote at that stage. Okay, so that one cell has two sources of information. Got 23 chromosomes from dad's sperm, 23 chromosomes from mom's egg, and now is a 46 chromosome zygote. And then that zygote, since it's one single cell, doesn't really make much of a human being, right? We got trillions of cells. It needs to undergo mitosis. So during cell division through mitosis, it's going to get bigger and bigger and bigger and become what we recognize as a human being. Okay, so that's our life cycle. And what we need to understand is that since we are diploid means two sets of chromosomes. We got one set from mom, one set from dad. So that means we actually have two copies of every chromosome. One we inherited from our mom, one we inherited from our dad. So we call those two copies together a homologous pair of chromosomes. So homologous chromosomes, they carry the same genes, but maybe not necessarily the same versions of the genes, but we have two copies. One we got from mom, one we got from dad. So let me show you what I mean in the next slide. Okay, so here is one chromosome. Remember, it's one chromosome. Even though we got sister chromatids here, it's held together. We have one centromere. Okay, so here's one chromosome. Here's another chromosome. Two centromeres. Okay, so we got two separate chromosomes. Now this chromosome carries the same genes as this chromosome. Okay, you got the pink gene, you got the orange gene, you got the green gene, but they came from two different sources. Okay, this one came from your mom's egg. This one came from your dad's sperm. So you have two copies of every gene. Okay, you got one from mom on that homologous chromosome. You got the other from dad from that chromosome. Okay, so you have basically a pair of homologous chromosomes. So chromosomes that are basically the same length and carry the same genes, they are considered homologous. So you got one copy from mom, one copy from dad of chromosome number 1. You got one copy from mom, one copy from dad of chromosome number 2. You got one copy from mom, one copy from dad from chromosome 22. Okay, so you actually have 23 pairs of chromosomes. 23 times 2 equals 46, so 23 pairs. So you have 2 copies of every chromosome. Okay. This, by the way, is called a karyotype. A karyotype is just a display of your chromosomes. So if I added up all of these chromosomes, I would get 46 of them. And you can see that you have 23 pairs. Okay, so here's, pair number 1, here's pair number 22, and then that's the 23rd pair. It's your sex chromosomes. In this case, this is a boy because there's one X chromosome and one Y chromosome. Okay, so you have 23 pairs of chromosomes. Okay. So here's our normal human karyotype with 23 pairs of chromosomes. So a normal human has 22 pairs of what we call autosomes. An autosome is a non-sex chromosome. Okay, so you have 22 pairs of them. That's chromosomes 1 through 22 are considered autosomes, and then that last pair of chromosomes is the sex chromosomes. So hey, guess what? They determine the sex. That's why they're called sex chromosomes. So for males, you have non-homologous chromosomes. For your sex chromosomes, non-homologous means they're not the same. You got one X and one Y. Females, you have homologous chromosomes. For your sex chromosomes, you got 2 X chromosomes. They carry the same genes. All right. So this is a karyotype of a male, and this is a karyotype of a female. Males and females look alike all the way through chromosome 22. It's just the last pair is different. Okay, so males have non-homologous chromosomes for their 23rd pair. Females have homologous. Okay. So getting back to the diploid versus haploid, there's your definitions for you. So each pair of chromosomes, homologous chromosomes, includes a chromosome from each parent. So if you're diploid, you have 2 sets of chromosomes. One set you inherited from your mom's egg, one you inherited from your dad's sperm. Okay, so 1 + 1 = 2, you got 2 sets. When you are a zygote, a fertilized egg. Okay, so where did that zygote fertilized egg? Remember, cells, they come from other cells. It didn't just poof out of nowhere. You were a zygote at one stage in your life where you were one single cell, and you didn't poof out of nowhere. You came from the union of your mother's egg and your father's sperm. Sorry. So we have the egg contained 23 chromosomes, or half as much as a zygote. The sperm had 23 chromosomes, or half as much as the zygote. So the sperm and eggs are haploid. They only have 1 set of chromosomes in there. So they only have a single copy of chromosome 1. They only have a single copy of chromosome 2. Okay? Whereas diploid, you got two copies. You got one from mom, one from dad. Okay? So there's haploid, think half as much. 1 single set of chromosomes, and diploid, 2 pairs, 2 sets of chromosomes. And then once we take that haploid sperm with its 23 chromosomes and unite it with a haploid egg with its 23 chromosomes, you get 46 chromosomes. It's diploid. It has 2 sets of chromosomes. And we call that fertilized egg a zygote. So zygote is just a single cell that has 2 sets of chromosomes inside that came from this union of sperm and egg, which we call fertilization. Okay. Now, once we have that zygote, that fertilized egg, it's diploid, right? It's 2N. It has 2 sets of chromosomes, but it's only a single cell. So the zygote will undergo mitosis because it'll produce more somatic cells to grow. So zygotes, they undergo mitosis to grow and grow and grow and divide and divide and divide and differentiate and all that fun stuff until they become human baby that's born, and then the grows into a child, and the child grows into a teenager, and then it grows into an adult. And then by the time it reaches adulthood, it has the ability to produce haploid cells. So through sexual maturity, those haploid cells will develop via meiosis, okay, because we can't undergo mitosis to produce gametes because then you have too many chromosomes in there. So the haploid cells are produced via meiosis, and they are produced in this a specialized area in your body. Remember, most of your body cells are somatic, but there's special cells in your gonads, which are the gamete-producing organs of your body. So for females, that would be the ovaries. For the males, that would be the testes. And inside the ovaries and testes, there's specialized cells, which we call germ cells. Those germ cells are diploid, but they undergo meiosis instead of mitosis. And so when they undergo meiosis, they're producing haploid sperm or eggs. Okay. All right, so let's take a closer look at that figure one more time. So there's that ovary, there's that testis, and inside the ovary and the testes, there's those germ cells that will, instead of undergoing mitosis, they undergo meiosis to take that 2N diploid germ cell and produce haploid gametes from it. So once we have our haploid gametes, we can have fertilization take place with a 23 chromosome sperm, haploid, and a 23 chromosome egg, haploid, to unite under fertilization to form a diploid zygote. So that diploid zygote has 2 sets of chromosomes now, one from the sperm, one from the egg, and it will undergo mitosis to grow into an adult. Okay. All right, so we've talked about meiosis is the production of those gamete cells, but how exactly does that occur? So we're going to go through meiosis, and just to give you an overview of meiosis, it's different from mitosis in the sense that in mitosis we're just making another skin cell, right? We're just making a duplicate copy. So meiosis is different because we're not only creating gametes, which are different from the germ cells they came from, and you got like an egg or sperm that look different. They don't have as many chromosomes in them. They only have 1 set of chromosomes instead of 2. And because of that, meiosis is going to undergo a series of cell divisions. It actually goes through 2 sets of cell divisions. It goes through meiosis part 1 and meiosis part 2. And in the end, instead of having 2 daughter cells like in mitosis, we end up with 4 daughter cells. And those 4 daughter cells will be the sperm or the eggs. Okay, so we're going to do a brief overview of these 2 cell divisions, and then we'll get into the nitty-gritty. So these 2 cell divisions, meiosis part 1 and meiosis part 2. In meiosis part 1, we're going to separate the homologous chromosomes from each other. So remember, how many pairs of chromosomes do you have? Okay, you have 23 pairs, right? So you have your homologous chromosomes, you got 2 copies, one from mom, one from dad. So you got 23 pairs of chromosomes. So in meiosis part 1, we're only separating the homologs from each other. The sister chromatids stay together. We'll get into that. Okay, so at the end of meiosis part 1, we're only separating the homologs from each other. In meiosis part 2, we're going to actually physically separate the sister chromatids from each other. So if you're confused, don't worry, we'll go through it. Okay, so here's an example. Here is interphase. This is before the S stage of interphase. So imagine this is, G1. We got, in this case, we have a pair of homologous chromosomes, and they color-coded them. So the blue one probably came from dad. The red one came from mom. So you got your 2 copies. You got one from mom, one from dad. So this is a pair of homologous chromosomes. What are homologous chromosomes again? They carry the same genes, but not necessarily the same versions of the genes. So, you got 1 copy of the genes from your dad, 1 copy from your mom. Okay, so these are homologous. And before we begin meiosis, we got to go through interphase, right? So S stage of interphase, what happens? DNA replication, and now you got, hey, copies, right? So we have 1 centromere, 2 centromeres. So we have 2 chromosomes here, we got 2 chromosomes there, but in this case, these 2 chromosomes, okay, they have the sister chromatids attached. So we have our sister chromatids held together by the centromere. These are duplicates. Okay, they're identical to each other. The sister chromatids are identical. This guy has its sister chromatids held together by the centromere, identical. The first thing we're going to go through is meiosis part 1, which is an entire cell division. And so in meiosis part 1, the only thing we're doing is we're separating the homologous chromosomes from each other. Okay, so these guys get separated and into separate cells. So now we have 2 cells at the end of meiosis part 1. But look, they're sister chromatids still stuck together. So we're not done yet. Then we undergo meiosis part 2. And in meiosis part 2 is when the sister chromatids finally separate. Okay, so we gotta, we still have duplicated chromatids held together here. So that's why we have to have that second cell division. And at the end of the second cell division, we end up with 1, 2, 3, 4 cells. And look at the composition of the cells. There's only 1 chromosome inside. Up here, there are 2 chromosomes. So we halved the amount. Haploid, okay? So let's start out with a video. Make sure we understand the visuals, and then we'll get into the nitty-gritty.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Okay, so that was a good overview of meiosis. And again, you got to pay close attention to the chromosomes in meiosis, okay? So, you can follow one single sister chromatid and follow it through and see what's happening. So let's take a closer look at meiosis part 1 and meiosis part 2. Now, meiosis will follow the same progression of phases like mitosis. So remember, in mitosis, we had the PMAT from the Amoeba Sisters video, prophase, metaphase, anaphase, telophase. Meiosis has that same order of events, it just happens twice. So we go through prophase part 1, metaphase part 1, anaphase part 1, telophase part 1, ending with cytokinesis, splitting the cells into 2. And then those 2 cells go through prophase part 2, metaphase part 2, anaphase part 2, and telophase part 2. And then each individual daughter cell divides into 2 more cells. So that's where we get the 4. So if you remember what happens in prophase, metaphase, anaphase, telophase, it's going to be very similar, okay? The only difference, though, is we have to pay close attention to the exact chromosomes, okay? So let's start with prophase part 1. So prophase part 1, we have very similar events to regular old mitosis prophase. We got the centrosomes, they start separating and migrating to opposite poles. We got the mitotic spindle forming from the asters of the centrosomes. And that mitotic spindle starts to attach to the kinetochores on the chromosomes. The only real difference, though, is take a look closely what's happening inside the nucleus before the nuclear envelope dissolves. So you see that instead of just the sister chromatids kind of like flying around nowhere, they're hanging tight to their homologous chromosome mate, okay? So we have a pair of chromosomes. Remember, those are homologous chromosomes. They carry the same genes. And so in this pair of chromosomes, you notice that we still got the sister chromatids, so we actually have a pair. There's 2 chromosomes here, okay, because there's 2 centromeres. But each chromosome has a copy because they're sister chromatids. Now, notice how they're hanging out together. Okay, so that's one key difference between mitosis and meiosis part 1. The homologous chromosomes are hanging out together. And because they're hanging out together, we have the possibility of something called crossing over occur. So let me just take this picture and kind of blow it up for you. There it is. Okay, so here is what we call a tetrad. So tetra means 4, right? So we actually have 4 sister chromatids on 2 separate chromosomes. There's one chromosome, here's another one, and they're homologous, so they carry the same genes. Okay, now they're so close together that look at what happens. We might have a crossing over occur, where a little piece of one of the non-sister chromatids breaks off and attaches to the other one. Okay, so look at what's happening with this particular sister chromatid. It has some of the red, but it also has some of the blue attached. Okay, so now this sister chromatid is different from that one and that one. They're unique, okay? That'll help with our genetic variation when we get into the gametes. But I just wanted to point that out. So this is occurring during prophase part 1. We get a little bit of a sister chromatid overlapping, and that can add some genetic reshuffling. Okay, so let's go back here. Okay, and you can see there's some genetic reshuffling there. We got a little bit of blue on the red and so forth. So at the end of prophase part 1, we have attached the mitotic spindle to the kinetochores. Everybody's hanging out in their homologous pairs. They're all friends. Happy, happy times. Then we're going to go into middle metaphase part 1. And it's going to be the same as in mitosis for the most part, where they get pushed and pulled. The only thing is that they're hanging out together. in their homologous pairs. Okay, so there's still the homologous pairs are hanging out together in that little tetrad. Remember, tetra means four. Okay, in anaphase part 1, the difference between mitosis and meiosis part 1 is that remember how in mitosis the centromeres dissolved and then the sister chromatids separate? Not in meiosis part 1. Okay, so in meiosis part 1, the centromeres stay intact. And so the only thing that's getting pulled to opposite pole is an entire sister chromatid pair. Okay, so the homologous chromosomes, they separate, but the chromosomes themselves with the sister chromatids, they stay together. Okay, so here's one chromosome with its sister chromatids pulling to one side. Here's another chromosome with its sister chromatid pulling to the other side. Okay, and then telophase 1 is, just like mitosis telophase. So you have the nuclear envelope starts to reform a little bit. But you notice what's happening inside this this cell. You have the two nuclei, but look at, you got sister chromatids still attached. Okay, now at the end of telophase, we're going to have telophase part 1. We're going to have cytokinesis. So this is going to divide into 2 cells. Now here's a question for you. Is this cell down here, is it going to be diploid or haploid? How many sets of chromosomes are inside? Are there 2 sets or 1 set? Okay. We started out with 2 sets. Here's one pair. Here's one of the pair. Here's the other of the pair. So we have 1 pair of chromosomes, right? There's 2 of them. At the end, it's only 1 pair. 1, I should say. So of this pair of chromosomes, we only have 1 copy. So we've gone from diploid to haploid. So we are haploid, but we're not done yet because we still got those sister chromatids attached. Okay, so we've gone from diploid to haploid. So the homologous chromosomes has separated. Okay, so here is another view of it. Here we have our beginning of meiosis. So you have the pair of homologous chromosomes. This is a tetrad. You got 2 separate centromeres, so you got 2 chromosomes, but they're a homologous pair. During prophase part 1, that pair, since they're so close together, they're intertwining, and so you can end up with something called crossing over, where a little piece of one of the sister chromatids breaks off and attaches to another. And so they're recombined. We actually call these recombinant chromosomes. And so at the end of meiosis part 1, we're going to just simply separate the 2 homologs from each other. So this one's going into one cell, this one's going into the other cell. At the end of meiosis part 2, we separate the sister chromatids from each other. Okay. So let's take a closer look at that crossing over effect that's occurring here. So I got a video.
Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here, the process produced a total of 4 genetically different gametes. See the different outcomes of crossing over. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent assortment and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Observe the combined effects of independent assortment and crossing over. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it's possible for for a human being to produce an almost infinite variety of gametes.
Okay. So that hopefully explained a little bit about that crossing over effect. And the outcome is extreme because it's providing genetic variability, which means that, you know, in addition to all of this genetic variation, each one of the sperm and eggs that you're producing are genetically unique. Okay, so that's adding to that variability amongst our offspring. Okay, so meiosis part 1 is a little tricky because we're basically just separating the homologous chromosomes from each other, which can get a little confusing. Meiosis part 2 is a piece of cake. It's basically just mitosis, okay? But you got 2 cells instead of 1. Okay, so in here is meiosis 2 prophase part 2. So we have 2 cells, right? Because at the end of telophase part 1, it divided into 2, so you got 2 cells here. So in prophase part 2, it's just like prophase in mitosis. So the nuclear envelope disappears, the mitotic spindle starts to attach to the kinetochores on the centromeres of the chromosomes. In metaphase part 2, same as in metaphase and mitosis, the sister chromatids, they get pushed and pulled until they are in the center of the cell. And then anaphase part 2, just like in mitosis, the centromeres finally dissolve, and that allows the sister chromatids to get pulled to opposite ends of the cell. And then telophase part 2, same as in mitosis, so you end up with the nuclear envelope reforming, you got the mitotic spindle disappearing, you get cytokinesis again. But the only big difference between mitosis and meiosis part 2 is look at how many chromosomes there are. There's half as many. It's haploid instead of diploid. And you got all these unique combinations of your genes. You got a little blue here attached to the red. You didn't have that in mitosis. Okay. In plant cells, it's a little different in the sense that you can see this is one single cell that has 4 daughter cells forming. So the cell plate kind of ends at the end of telophase part 2. All right, so let's do a similarity and a differences between mitosis and meiosis all in one slide. So let's start out with a single parent cell. Now let's see how many chromosomes this parent cell has. Count the centromeres. 1, 2, 3, 4. So there's 4 chromosomes here, and it's a diploid cell. So it has 2 copies. So we have 2 copies of one chromosome, 2 copies of the other. So the first thing we do is we undergo DNA replication during the S stage of interphase. So both of them do that. You got your sister chromatids. We still have 4 chromosomes. 1, 2, 3, 4. 1, 2, 3, 4. Okay. But then here's where the differences come in. In prophase part 1, notice the homologous chromosomes are sticking together instead of just floating around as in prophase. Okay, so the homologous chromosomes stick together so much that they can end up with the crossing over effect occurring, and you get some little genetic variation shuffling there. In metaphase part 1, here's another difference. Mitosis, they're just lined up. In metaphase part 1, they're lined up as homologous pairs. And then anaphase and telophase, big difference here. In anaphase and mitosis, the sister chromatids separate, and you end up with your daughter cells. In anaphase part 1, the sister chromatids are stuck together. So you still have the sister chromatids together, it's just you're separating the homologs from each other. At the end of telophase in mitosis, you still have 1, 2, 3, 4 chromosomes in each daughter cell, and it looks just like the parent. At the end of telophase part 1, in meiosis, you got only 1, 2 chromosomes. You got half as much as the parents, and those sister chromatids are still attached. So you got to undergo meiosis part 2 to move them apart. So at the end of meiosis part 2, you have 2 separate, 2 chromosomes in each cell, so half as much, and you got 4 cells instead of 2. So they're haploid. Okay? And that is an important difference because these cells, they're genetically unique and they only have half as many chromosomes, so they're going to be used in sexual reproduction. These cells over here, they are the exact copies of the parent cell, so they're going to be used to basically for growth, tissue repair, and asexual reproduction. All right. So here's a good difference between or comparing and contrasting mitosis and meiosis. So remember, mitosis and meiosis, they are occurring in eukaryotic cells, not prokaryotic cells. Prokaryotic cells, they don't do either. They do binary fission. As far as the function of mitosis, it undergoes growth, repair, asexual reproduction. For meiosis, it's sexual reproduction only. In mitosis, you're making genetic clones, okay, so it's genetically identical. Meiosis, those gametes are varied. Okay. At the end of mitosis, you have 2 diploid daughter cells. At the end of meiosis, you have 4 haploid daughter cells. Okay, so remember, haploid means half as many chromosomes. So you have 1 set of chromosomes instead of 2. Okay. In mitosis, you have both mitosis and meiosis undergo the S stage of interphase, so that's where the chromosomes replicate. But in mitosis, you only have 1 cell division, whereas in meiosis, you have 2. Okay, and then finally, other characteristics. We have that in meiosis, you have that crossing over effect occur with the tetrads coming together, and that increases the genetic variation. Okay, so one important thing that results from meiosis is genetic variation, right? So every single gamete that is being produced has a unique combination or unique shuffling of the parent's genes. And that is due to a couple of things. It's due to the crossing over effect, which we talked about, occurs during prophase part 1. It also occurs due to the independent assortment of chromosomes. Now this occurs excuse me, during metaphase part 1, where the homologous chromosomes, they're hanging out in tetrads, right? And they align themselves kind of independently of each other. So we're not going to always have, one maternal set on one side and the paternal set on the other. It's going to be kind of shuffled around, and we'll talk about that. So these 2 things add to genetic variation during meiosis. And then finally, random fertilization is occurring that adds to even more genetic variation. So each sperm has the same chance of fertilizing each egg, okay? So it's just randomly. The egg doesn't choose, I like that sperm more than that one. Nope, it's completely random, right? So that all adds to genetic variation. So let's watch an animation to demonstrate this.
Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we've color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you can see one of the possible arrangements and outcomes. Let's rearrange the chromosomes to get different arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent assortment of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being with 46 chromosomes, more than 8 million combinations are possible.
Okay, so you can see how we have the crossing over effect, but you can also see this was the independent assortment of chromosomes. So in that case, the paternal ones are kind of all on one side and the maternal ones are all on the other. But over here, you get like a little change there. Okay, so that's that law of independent assortment that I told you about. Now this is a cell that only has 4 chromosomes. Well, we have 46, right? So there's even more combinations than that. Okay. So that independent assortment of chromosomes occurs where during metaphase part 1, the homologous pairs randomly assort themselves at that metaphase plate. So each pair of chromosomes sorts the maternal and paternal homologs independently of each other. So we're not going to have all the mommy's chromosomes on one side and daddy's on the other. It just kind of independently assorts that way. And so that leads to a number of different combinations possible. And the way we figure that out is we use this formula. So 2 to the nth power. The n stands for the haploid number. So what's our haploid number? Our diploid number is 46. So our haploid number is 23. We have 23 chromosomes in our sperm and egg. So if I do 2 to the 23rd power, that makes it so that I have more than 8 million possible combinations of my chromosomes. That's a lot, okay? And that's just from that independent assortment of chromosomes. I'm not even talking about crossing over or random fertilization. So just that one step of independent assortment of chromosomes, I have 8 million possible combinations of my chromosomes, okay? So that's a lot of possibilities. So just to show you a little bit closer, so here's one possibility. So I got the of my 4 chromosome cell here. We got the blue ones on one side, the red ones on the other. So that means that they're going to separate and segregate. Okay, so now I only have, these kind of possibilities. Let's say that the arrangement was a little different. So instead of blue on one side and red on the other, I got like a little differences there. Okay, and here's another combination. So I have 4 combinations from this set. But this is just for a 4 chromosome cell. I have a lot more than that in my cells. I got 46. So there's over 8 million combinations of mine. Okay. So independent assortment of chromosomes is one way we get different daughter cells, different gametes. And the other way is through crossing over. Okay, so remember crossing over, you have that crossing over effect that occurred. And so you're going to end up with a unique combination of the parent's genes on the individual sister chromatids. So we call those recombinant chromosomes because they're recombined. They have unique combination of both parents' genetic information. In humans, we can have 1 to 3 crossing over events per chromosome pair. That's a lot because we got, 46 or 23 pairs, right? So, I mean, if we have 1 to 3 crossing over events per pair, then that means that we have a lot of reshuffling occurring, lots of recombinant chromosomes taking place. Okay. So here is that we have our tetrad where we have our homologous chromosomes so close together that they kind of flop over each other. And as they flop over each other, that crossing over can occur. And then we end up with that recombinant chromosome there. So now we got, these chromosomes that are all different from each other, genetic variation. So that's another way that adds to our genetic variation. So we talked about the independent assortment of chromosomes and that crossing over effect. And then finally, we have our random fertilization. So in random fertilization, any sperm can fuse with any ovum or egg, okay? So each sperm and egg contains about 8.4 million possible chromosome combinations due to that independent assortment of chromosomes. And then, you got 2 random gametes fusing together. So that means that, 8.4 times 8.4, that's over 70 trillion diploid combinations that can occur. And that doesn't even include the crossing over effect. So basically, when it comes down to it, every zygote is unique, okay? So even if we have siblings, you came from the same parents, you're a unique combination of your parents' genes because you came from different gametes. The exception, though, is identical twins. In identical twins come from one single zygote. So one sperm fertilized one egg, you got one zygote. And then early in that embryonic development, that zygote split into two different humans, okay? So that's where identical twins come from. In that case, they have the same basic genetic combination of their genes, okay? So that's why they look the same, okay? But environmental differences can cause some changes. Unless you have an identical twin, nobody out there has the same genetic combination as you, okay? So even your siblings. All right, so we'll go ahead and stop right here for today. That concludes chapter 10. So you do have an exam. So your second midterm exam covers chapters 6 and 7 and 9 and 10. Don't do 8 because that's photosynthesis. So what did we cover in 6? That was enzymes and cell signaling. In 7, we went through cellular respiration. So basically, those are the functions of the cell. 8 and then 9 and 10, we went through cell division, mitosis and meiosis. So you should have a good understanding of not only how the cell functions, but how it's dividing. Okay, so if you have any questions, feel free to email me, or you can post a question on the student Q&A. Otherwise, yep, you should be good to go now for your midterm exam. So we've covered everything for the midterm exam.